Hello everyone. Welcome to part 5 of the lecture series. So this is, I would say, one of the most important and fundamental lectures of the series. It talks about the important concept of central dogma and it describes all the basic concepts around. So we are going to discuss how DNA transforms into RNA and how RNA transforms into protein in this lecture. So the first thing is, even before we talk about how DNA transforms into RNA, we should actually understand how DNA itself is created. So we have a few DNA, and obviously our cells replicate, our cells grow, and we grow as humans. So how do DNA grow? How do DNA actually get, I would say, produced from the mother DNA? So the process of how a new DNA is produced from the mother DNA is called DNA replication. So it's actually a very simple process. You can see that there is a double helix DNA right here. This is, you can think of this as the original or mother DNA. Now what happens is this double-stranded DNA breaks. So you can see that the strands have been broken. So there is one strand and there is the other strand. So when DNA actually breaks into two single strands, now human body is designed in such a way that it doesn't allow DNA to stay in the single stranded form for a very long time. So what happens is the DNA polymerase enzyme that is there in our body, it starts repairing those, what should I say, single strands. So for this single strand, it keeps attaching the complementary bases. So for example, if this is A, this will be T. If this is C, this will be G. If this is G, this will be C. You know about complementary bases, right? From lecture 1. So it will do that for this particular strand. And similarly, for this strand, again, same DNA polymerase enzyme will create the complementary bases. If you look closely, this is the most interesting part. This particular new DNA, new double helix DNA, is exactly the same as the mother DNA. Similarly, this particular new DNA, child DNA, is again exactly the same as the mother DNA. Why is that? Because the complementary bases are going to be exactly the same, right? Because we know what the complement should be. That is why the child DNA, both the children DNA are exactly the same as the mother DNA, right? But there is one thing. Sometimes what happens is if your body is going through stress like cancer or things like that, then this replication process can have a few errors and that can create some problem in your body, but not in normal cases. Okay, now that we are done with DNA replication, let's now focus on the central dogma. So central dogma is actually associated with three processes. One is the DNA replication that I just described. Then from DNA to RNA transcription, RNA to protein translation. So there are two things, transcription and translation. So remember these two names because if you're working with bioinformatics, you are going to hear their names again and again and again. So I don't think anything describes this process better than this image. So first of all, I will describe the transcription part where a part of DNA, maybe this part, transforms into RNA. So as you can see in this image, the DNA is double helix, double stranded, but RNA is single stranded. So I will describe everything now about DNA to RNA transcription. So first of all, you can see that this is a DNA, double stranded DNA, and you can see some names written here, exon, right? So this is the first time you are seeing exon, right? So exon is also known as coding regions of the DNA. So this particular word gene, this is known to you, familiar to you, because in the last tutorial on epigenetics, we talked about gene, right? We talked about some gene regions which are actually responsible for almost all functions of our human body. When the gene gets activated, some functions of our body is performed. So the gene regions are actually consisted of exons. So multiple exons can actually create one gene region. So that's the basic thing. Now, exons, although here you can see a lot of regions are actually exons, in reality, only 1 to 2%, I will repeat, only 1 to 2% of human genome is covered by exons. So exon region is very, very limited. And there is actually a sequencing called deep exon sequencing. So previously we talked about whole genome sequencing, and also there is one thing called whole exome sequencing. So since exons are actually very few in number, whole exon sequencing are normally very deep. So you are going to do like 100x or 1000x deep sequencing in the exon regions to understand them more. So it is very common. So again, I will repeat the name. It's called whole exon sequencing. Okay. Now, so what happens is in the process of transcription, this double strand actually gets transformed into a single strand, and this is called a primary RNA transcript. This is called a primary RNA transcript. Now, from this primary RNA transcript, something called mRNA is created. Now, obviously, you are thinking that, I mean, What is mRNA? I mean, is there other types of RNA? Yes, it is true. There are other types of RNA, but mRNA is the most crucial one, so I will talk about this first. So mRNA, M is for messenger, so it's a messenger RNA. So from this RNA transcript, through the different combinations of exons, different mRNAs are created. So understand that from this single gene region, which consists of multiple exon regions, we have a RNA transcript, and from this single RNA transcript, we can actually create different mRNAs through different combinations of the exons. And ultimately, this mRNA actually translates into protein. Okay, so that is the very basic concept. Now, this process of randomly combining these exons is called alternative splicing. Another terminology alert. So it is called alternative splicing. Okay. Now that we know this much, let's also talk about the two other RNA types besides mRNA. Their names are very simple. tRNA means transfer RNA and rRNA means ribosomal RNA. They are also created from the DNA itself, but they are created from non-coding DNA regions. Okay, they are not created from coding DNA regions. They are created from non-coding DNA regions. I also want to mention one more thing. You can see a lot of spaces in between, and actually there are a lot of spaces because only 1% or 2% of the entire genome is exon. So those places which are not coding, which don't have actually, we don't really know a lot about their functions. Those regions are actually called intron. Okay, intron. Intron, intron. Okay, and now that we know that, obviously one question is coming to your mind. If we are focusing on mRNA, if mRNA is making proteins, what is the function of tRNA and rRNA? They have very important functions and they are very intuitive. I will talk about them in the next slide. But before that, let's look at the basic structure of RNA. So this is how an RNA looks like in 3 dimension. Now, obviously, you can actually tell me that you are saying, Rafid, that this is single-stranded. So why does this look double-stranded? Actually, this is also a single strand. It started from here and it coiled around a lot and then ended here in the 3' end. So now the next question is, why are these bases connected, right? Yes, RNA bases can be connected in some places, but they have nothing to do with double strand. A single strand can connect its own base with itself when it coils around in the 3 dimension. Okay. So this is one thing. Another important thing is there is no T in the RNA. So as you know from before, in the DNA we have A, T, C, and G, right? But in the RNA we have A, G, C, and U. So there is no T. T is replaced by U, uracil. Okay, so that's the only difference between RNA and DNA. Okay, now let's talk about a little bit more. So obviously you can understand that RNA is also a sequence. It's a sequence of bases. So let me see if I have my pen somewhere right here. So you can think of it as a sequence of maybe A, C, G, G, U, C, etc. Right? So 3 consecutive bases are called codon. So this is one codon. This is another codon. Now the question is, why are they called codon, right? Actually, codons have very important implications because codons are the one which ultimately transforms into amino acid. So each codon, each three consecutive bases of an RNA converts into amino acid. I know that a lot of things are being thrown at you because there are a lot of things here. So what is amino acid? Amino acid is basically the building block of protein. Okay. So let me actually show you this. So, I mean, there is more in the next slide, but before showing this, let me show you this thing. So you can see these are different amino acids, and actually there are 20 unique amino acids, 20. Okay, 20 unique amino acids. So let me write something again. So let's say we have a sequence like this: A, C, R, A, G, N, and so on and so forth. So this sequence of amino acids, which are represented by these letters, I mean, there are more. I haven't written all 20 of these, and these are the full forms. So this sequence of amino acid makes up what? It makes up a protein sequence. Okay, so this is what protein is. Now, protein has different structures, which I will talk about in probably part 7. So I will not talk about that. But this is the basic structure of protein, and in many, what should I say, many files, sometimes, I mean, these files are known as FASTA file, I mean, dot FASTA file. So you have dot FASTA file. So in this FASTA file, you can actually see these sequences. Like you have these long strings of A, C, R, these 20 amino acids, and those are basically proteins, how the proteins are stored. Okay. Now we basically understand that each codon, that means 3 consecutive bases turning into amino acids. And amino acids make up the big protein chains. Fine, that is very good. But now let's understand a little bit more. Let's actually go further. This is very important to understand that a codon consists of 3 places, right? 3 placeholders, because 3 consecutive bases make up a codon, right? And a codon transforms into amino acid. So understand that we have 4 things here: A, C, G, and U, right? That means any 4 of these letters can actually take this place. Any 4 of this letter can also take this place. Any 4 of this letter can also take this place, right? That means we can have a total of 4 cube or 64 combinations of codons. So there are 64 possible unique codons available. But understand that codon is transforming into amino acid, right? Amino acid. But the issue is we have 64 different codons, but we have only 20 amino acids. What does that mean? That means there are multiple codons which transform into the same amino acid. So the mapping from codon to amino acid is called a many to one mapping. Okay, so take a moment to grasp this concept, and I will now show you a chart where I will have different codons and mapped to the amino acids. Then it will be even clearer. So let me do that, I think, now, because now is a good time to do that. So let me go to the previous slide. Okay, let's look at this particular chart first. You can see that there are 64 possible codons. You can see these 3 letters. All of these are codons. So there are 64 of them. You can count them up. And you can see that some of them actually transform into the same amino acid, LEU. Okay. So I think how many? 6 of them transform into the same exact amino acid called LEU, right? So there is no change. There is absolutely no change in amino acid whatsoever, in spite of the codons being different. That is why it's called many to one mapping, right? Okay, enough of that. Now I will talk about how exactly this RNA, or I should say mRNA, messenger RNA, as I showed you right here, translates into protein. So let's look at this picture. So from this DNA, we have this place which is the gene or the exon, and it transforms into an mRNA. This is the mRNA. So this is the blueprint of making the protein. Now this blueprint is actually carried by something called tRNA. So these are transfer RNA. They carry this mRNA information to something called rRNA. So this egg-shaped thing, this is the rRNA, ribosomal RNA. So it takes the transfer RNA, transfer information from mRNA, and it gradually makes the entire protein. Okay, it gradually uses this information to make the entire protein. That is exactly how the protein is made, through a growing chain of amino acid, right? That is actually it. I mean, it's not obviously as simple as I'm making it, but for bioinformatics guys who are mainly going to do computational stuff, right, or studies, for them this is good enough. So that's basically how the entire dogma, I mean, going from DNA to RNA to protein works. So hopefully you enjoyed the video. So in the next lecture, what I'm going to do is I'm going to talk about gene expression and how exactly gene expression is measured. So when we are talking about DNA to RNA transcription and protein making, it's basically a form of gene expression. So what does gene expression mean? Gene expression means your gene transcribes to some RNA and that transcribes to some protein. So we are going to discuss this concept in more detail in the next video, and in the subsequent video I'm going to talk more about the protein structure, because here I just talked about how the protein is made, but there are a lot more to that, I mean, in proteins. So I will talk about that as well. So see you in the next videos. Thanks very much.